So, welcome back. Yet another unit review video. This is actually the last one I'm going to be making. We still have units 9 and 10 on the curriculum, but those are the evolution topics that we're currently working on. And it seems kind of silly to make a review video about the topic that we're in at the moment. So this will be the last one. This is unit 8, genetics 2. And the first prompt that I want to look at is the one that asks us to model meiosis and describe its products compared to its original starting cell. Okay. So meiosis is the cell division type that we utilize to create the gametes that we need to do sexual reproduction. So those are cells like sperms and eggs. And now, as I'm sure you know, the thing about sperms and eggs is that they have only half the total DNA of the original organism. Right? That's so that in order that when they come together through fertilization, they can make one whole, half whole. Okay. And so meiosis is the process that our body utilizes to generate those gametes with half of the DNA. Specifically, it's a process of separating the DNA into that half set. Now, we, like a lot of organisms, start as a diploid creature, right? Meaning that we have 2 copies of every type of chromosome that we have. Now, as humans, we have 23 types of chromosomes. We have 2 copies of each, meaning that we have 64 distinct chromosomes in our genome. Yes, no, is that math? Mr. Lavadier, you goon. It's 46, not 64. I reversed it. 23 times 2 is 46. So we have 46 types of chromosomes, distinct chromosomes, right? Because we have 23 types. In my model here, I have 3 types of chromosomes. I have chromosome type A, chromosome type B, and chromosome type C. And I've differentiated between the maternal and paternal set of those through color. And so the green is the paternal set, the blue is the maternal set. So you can see that there's 2 A's, 2 B's, and 2 C's. They're the same type of chromosome, but they're slightly different in their allele sets. And hence the same letter, but different colors. I've chosen to represent this as being 3 sets times 2 types, right? 3 types times 2 copies, being diploid. Our cells go through S phase, replicate the DNA that they have. So each of the chromosomes gets copied. So now I have 2 copies of each of the 3 types with 2 copies. And so if you look in the model here, it's a little bit clearer than what just came out of my mouth. So the A, the green A, now has 2 green A's and 2 blue A's. I now have 2 blue B's and 2 green B's. 2 green C's and 2 blue C's. Right? I've doubled up everything I have. I haven't added any new information in here. I've just doubled what I already had. Okay. So it's still diploid. Now meiosis begins, and the cell goes through PMAT1, prophase 1, metaphase 1, anaphase 1, telophase 1. And what that does is separates the homologous chromosomes from one another. Right? And so my A's get separated. So my green A's end up in one of these daughter cells, and my blue A's end up in the other one. Same with the B's, and same with the C's. Okay, that's what PMAT1 does. At this point in time, we now have a haploid organism because it only has 1 type of each of the chromosomes, right? I only have 1 A type, I have green A. I only have 1 B type, blue B. But I have 2 copies of each, so I have 2 times the 3 types. These daughter cells are then in turn going through PMAT2, which is separating the sister chromatids from one another. And so we zoom in. So these 2 green A's got separated, one goes here, one goes here. These 2 blue B's got separated, one goes here, one goes here. 2 C's got separated, one goes here, one goes here. And down here, the same thing happened to this daughter cell. Okay. And so now we have a haploid cell that only has 1 copy of each of the 3 types. So if we compare this to the starting cell, we see that we've reduced the total DNA set by half, but we've kept the overall structure intact. Right? And so the types of chromosomes are A, B, C, and my gametes have A, B, C. They only have one copy of them. Okay? So that's meiosis. That's what's going on there. So the next section of bullet points from this unit that I want to look at is the one that asks us to address Mendelian inheritance. So Mendelian inheritance is that basic inheritance pattern that we typically think of, right, where we have one dominant allele represented by an uppercase letter and one recessive allele represented by a lowercase letter. Okay. Being diploid, we have two copies of each of these genes. And so there's three setups that we could possibly have for any given gene. And the setup, the combination that we have, we call the genotype. So if I have two dominant alleles, I call that homozygote dominant. If I have one dominant and one recessive, I call that heterozygous. And if I have two recessive alleles, I call that homozygous recessive. Okay? Those are our genotypes. Homozygote dominant, heterozygote, homozygote recessive. Okay? The phenotypes is what the actual trait is expressed as. Right? So if this is fur color and I have red versus blue, my phenotypes would be red fur or blue fur. Okay? A homozygote dominant organism in Mendelian inheritance presents as a dominant trait. A heterozygote organism in Mendelian inheritance presents as a dominant trait. A homozygote recessive organism in Mendelian inheritance presents as a recessive trait. We take the genotypes of the parents, we plug them into a Punnett square, which represents the process of meiosis, right? So this was separating this into its gametes, this into its gametes, and then fertilization is inside. So if this sperm fertilizes this egg, I would get this offspring. If this sperm fertilizes this egg, I get this offspring. This sperm, this egg, this offspring, this sperm, this egg, this offspring. Okay. And then we break that down into genotypic and phenotypic ratios. Okay. So the genotypic ratio of the offspring of this crossing is 1/4 homozygote dominant, 2/4 heterozygous, and 1/4 homozygote recessive. And now you notice that I did not simplify this fraction. Okay? Unless all fractions can be simplified to the same denominator, you leave them unsimplified. They should all have the same denominator. Over here, I have my phenotypic ratios based on these genotypes. So 3/4 of these organisms will present dominant, and 1/4 will present recessive. Cool? Which brings me to this last topic point of this topic review, the non-Mendelian inheritance patterns. So these are variations of that typical setup. So co-dominant non-Mendelian is when a heterozygous organism presents with both phenotypic options expressed. So if it's red fur and blue fur, if it's co-dominant, the heterozygous organism will be red and blue. Okay. Incomplete dominance is a very similar idea. However, instead of presenting together, the two versions present as a blend. Right? And so a heterozygous organism under the same conditions, red and blue, will present as purple. Because you take red, add blue, comes out purple. Multiple allele is simply that there's more than two alleles for that gene. That's it. 3 alleles, 4 alleles, 5 alleles, 142 alleles, more than 2 alleles. Sex-linked is that the gene that we're looking at is on either the X or the Y chromosome, the 23rd chromosome, the sex chromosome. That impacts inheritance patterns because males and females have different sets. Right? It's what's called allosomal chromosomes. They're not the same as one another. So the Y chromosome is structurally different than the X chromosome. The Y chromosome has less genes on it. So as males, we got XY. I have one X and one Y. Right? So I have one copy of every gene that's on the X chromosome. I have one copy of every gene that's on the Y chromosome. I do not have any genes to potentially cancel out whatever gene I have on my X chromosome, right? So whatever my mom gave me, that's what I got from my X-linked traits, whatever my mom gave me. That's it. Okay? And for me, as a male, I have Y-linked traits as well. Whatever my dad had for his Y-linked traits, I have for my Y-linked traits, right? I can't get a Y from my mom. My mom has no Ys. She's XX. So for Y-linked traits, I have what my dad has, and he has what his dad has. So therefore, I have what his dad had too, science, okay? Sex-linked traits. And the last one is polygenic. So many of our traits are coded for by more than one gene, right? They work in conjunction with one another to create the phenotype. Right? Skin tone, eye color, height, weight. Pretty much almost all of our traits are actually polygenic, right? And so very often polygenics works quite simply where the different genes, the proteins that they make just kind of stack on each other and lead to this gradation, right? So skin tone's a really easy way to see it, right? You have a bunch of genes that code for skin tone, right? And they can either be making melanin or not making melanin, because that's what gives our skin its pigment, right? Melanin. Melanin is brown, right? And so the more melanin I have, the more brown I am. Right? And so the more genes that are positive for melanin, the more melanin proteins I have, the more they stack up, the darker I get. And that's it. And it's pretty straightforward. Right? Clearly, I don't have many genes in the melanin side of things. I can't even get a tan. And so I'm lacking in the melanin category. Another way that polygenic traits can work is a little more interesting is epistatic traits, where there is your main trait, which, like, let's just pretend I'm a cat and I have fur. Let's pretend I have beautiful pink fur. Right? So I have a protein that's pink in my fur. And that's my primary gene controlling my fur color. I can have an epistatic gene that can mess around with my pink protein, right? And so I have this other gene over here, it codes for this protein. Maybe it globs onto my pink and it puts one little protein area up. And so now I'm sparkly pink instead of just regular pink, right? So if I have pink gene plus sparkly gene, I could be sparkly pink, right? But maybe I have blue gene, right? And my sparkly gene doesn't fit on my blue protein, so I can't be blue sparkly, which is sad. But I can be sparkly pink. Bye.